coming to the first International Women's Day seminar hosted by the Department of Life Sciences. First of all, I'd like to tell you, don't run away afterwards because we've got tea and coffee outside on the mezzanine afterwards. So it's a pleasure to invite Professor Anne Dell to give this first lecture. As you'll see from the notes, Anne first studied chemistry at the University of Western Australia, and she then was awarded a scholarship, an 1851 exhibition research scholarship to come and do her PhD in Cambridge. Subsequently, she moved to the department, what was then the Department of Biochemistry at Imperial College as a postdoc, and rose through the ranks to get a personal chair in 1991. She was head of department from 1999 to 2001, and amongst the honours, she was elected FRS in 2002, to the Academy of Medical Sciences in 2004, and in 2009 was awarded a CBE in recognition of her services to science. So Anne was one of the first people to use and develop mass spectrometry to study biopolymers, and in particular, she's led the field in application of mass spectrometry to study glycoconjugates. So her lab supports a wide range of labs, research groups throughout the world who are looking at the effect of glycans in health and disease. And so it's entirely appropriate that she delivers this first lecture, which is entitled The Sweet Things in Life.
Thank you, Barry, and thank you all for coming. I'll see whether this technology works. So let me start by telling you what I'm going to talk about. It's always a good way to start. So I'm planning to begin by telling you a little bit about my life. You've had a sort of dry summary of it. I thought I'd illustrate it with a few pictures. And then, because I know that probably most, with the exclusion of the people from my group who are here, of the audience, probably know very little about glycobiology, and so I do need to give you a bit of an introduction to why glycans are important. And then I thought I'd focus on two topics which have been part of my life for the last 20 years or so. Well, actually, one has been part of my life for the last 20 years or so. The other has been an aspiration of the last 20 years or so, which is immortality. Right. So, my background. So as Murray said, I did my undergraduate degree at the University of Western Australia. I actually grew up on a relatively isolated farm in Western Australia. So it was isolated purely because it was a little bit difficult to get to it with respect to infrastructure. It wasn't that far from Perth, if people are familiar with the capital city of Western Australia. But the farm that I grew up on, my father had bought the land during the Depression years, and soon after that, the state government had converted the whole of the area into a state forest, partly because it was the catchment area for a dam which provided water all the way across Western Australia to Kalgoorlie and Coolgardie, the eastern goldfields. So what that meant was that the government then didn't put any infrastructure into this particular valley, so there was no electricity, no running water, ironically, because there was a pipe that took water to the eastern goldfields, but not one we could tap into. And because we didn't have electricity, we didn't have what most people, even back then, it was quite a long time ago, were used to growing up with, radio, TV, etc. But also in addition to that, the children in the family didn't actually go to school for most of their early lives. I was actually lucky because I was the youngest, and I was allowed to go to school when I was 11, so I did have the whole of teenage schooling. But my older siblings mostly only had about a year or so of schooling. So I went to the University of Western Australia after completing high school, but I didn't actually stay at the university. Australia was a bit different from the UK. It was very unusual, unless you really lived in the outback, to board at university. People stayed at home and commuted to university. So I commuted from home, and this is my commute back in 1968 to 71 on my bike, going down the hills and up the other side. to the nearest town, which was approximately 6 miles from home. And it took, obviously, the length of time it takes to ride a bike 6 miles. You could go really fast in one direction. Bikes, we didn't have much money. The bike did not have anything more than the very basic 3 gears that bikes could have in those days. They weren't mountain bikes. So it was pretty tough going up the hills. So into Kalamunda, and then a bus down to Perth and a bus out to the university. So my commute each day was roughly 2 hours, lengthened actually by chemistry practicals. Those were the days when students never complained about anything and certainly didn't raise any issues you were grateful to be taught by your lecturers. And the fact that they scheduled the chemistry practical to run up to about 5:45 in the evening, even though university was meant to finish at 5:30, meant that I missed the commuter bus back from Perth, back up to Kalamunda, because Kalamunda was a small place, and they put buses on for the commuters, and they put buses on for people who were coming back from having fun in the evening. So the last bus left Perth at about 6 o'clock in the evening until people finished having fun, which was 9 to 10 o'clock, and then another batch of buses were put on. So what that meant was that I had to stay usually in one of the gardens in Perth for a couple of hours waiting to get the bus home. My youngest brother, who went to school at the same time as I did, he was a year older than me, he never complained, even though he was required to stay, even though he didn't do chemistry, he had to stay to make sure that his sister was home safely. So obviously it was quite late at night on the chemistry late practical days. In 1971, without my knowledge actually, my mentors, my honours year supervisor at the University of Western Australia, applied for a scholarship on my behalf, which is the scholarship that Murray referred to, the 1851 Science Research Scholarship. This is 1971 email. So the way you heard about things like this in 1971 was via a telegram, or telegraph, as is the word written up there. So the Great Exhibition, I would expect, talking to an Imperial audience, you are, I would imagine, sort of know what it is, or at least have heard of it. Quite a remarkable exhibition if you're not aware of the details of it, because it was held, obviously, a long time ago, at the time when trains were literally had only about a decade earlier started running across the UK from Manchester, Bristol, elsewhere to London. And yet a third of the population of Britain came to see the Great Exhibition, roughly 6 million people in 1851. And that was in a very short time. The exhibition opened in May of 1851 and closed at the end of September. And during that time, all these people came to see it. The other amazing thing about it was that it made money. That is money in 1851 terms, which obviously is somewhat inflated today. And that money was used to buy all the land to the south of Hyde Park, and obviously the land that Imperial College sits on today. And in fact, Imperial College pays a peppercorn rent to the Royal Commission for the exhibition of 1851, right up to today. In addition to starting essentially all of the stuff that's currently here, some of the money went to grants and scholarships, primarily at that time for bringing people from the British Empire to study in the UK. So I was very fortunate in being awarded one of these, and it did literally change my life. Not quite like that, although I have to say that going from having lived on an isolated farm, never spent a night away from home in the whole of my life up to that point, being transported from Perth on a plane, which I didn't really know how to catch or fly on, over 2 days, which is what it took in those days to get to the UK, was a bit of a shock to the system. I was fortunate, I think, in choosing, even though I didn't really have any advice on it, a college in Cambridge, which was a very forward-looking college. I had thought it would be very traditional, but in fact it wasn't. It had already cancelled things like May balls because the college thought they were elitist, and had introduced a way of life within the college, which was not really quite Cambridge-like at the time. There was no high table, for example, if people were familiar with that aspect of college life. The other thing about King's College was that I was lucky or unlucky, depending on one's point of view, to be the first woman in King's College, Cambridge. Now, this was a complete accident. First, with respect to the timing. So King's College, amongst half a dozen Cambridge colleges, went co-ed for the first time in 1972, the year I arrived. And I was arriving for the 1972 academic year to start my PhD. However, because the 1851 didn't give any travel money, and I didn't have any travel money, I had to apply for a travel award. And I was eventually fortunate to get an award from the British Council. But that took time. It took about 6 months. And so I arrived in Cambridge in June of 1972, thinking I was very late. What I hadn't realized was the academic year 1972 started in October. So when I arrived in Cambridge and went to see the person who'd been arranging my accommodation within King's College, I hadn't, of course, said I was arriving, because you didn't in those days. You might send a letter perhaps months in advance. So I obviously didn't give the exact date and time. And this guy was totally horrified. He said, "We haven't got any accommodation for women. We're not taking women until October. We can't give you a room in college because we don't have anything that's suitable for a woman." So I must admit I wondered, without asking, why you needed something different from the rooms they already had. And went rather very sort of sadly. I was incredibly sort of... well, I was homesick and miserable, I have to say, for my first few weeks in Cambridge. But someone had given me an address in London that actually in Richmond, that I had been staying at for a couple of days. So I went back there, and they said, "Wait to hear from us." And they came to a wonderful Cambridge compromise, which was that, no, I couldn't have a room in college, because obviously they weren't suitable for women, but I could stay in one of their houses on the backs of the River Cam. Many of those houses, as you may know, if you're Cambridge people, are owned by the colleges. And the reason I could stay in it was because it was the summertime, there were no undergraduates, and it was empty. So that was okay. I could stay in this empty terraced house. Soon after that, I came, incidentally, to work on an NMR project. So that had been the other thing that I had been rather sad about soon after I arrived, when the people in the lab that I was meant to be joining in Cambridge sort of gave me the news that my supervisor, who at that point wasn't there, because it was summertime, that he was planning to relocate to the University of Geneva, that he had taken up a professorship at the University of Geneva. Hadn't told me that. So when he returned, he said, obviously, yes, you know, you can come to Geneva with me. And I said, I don't really want to come to Geneva. I can't speak French, and it's bad enough coming to Cambridge. And in addition to that, I have an 1851 award to come to the UK. Now, fortunately, Howard Morris had been a postdoc with my putative supervisor until about a year before, when he had been given an appointment at the Laboratory of Molecular Biology in Cambridge, but the chemistry department, where he was based, had allowed him to stay in the chemistry department. So Howard had come back from his holiday, and not only did he offer to be my supervisor, but he also, very kindly, he was married with a small child at that point, said that they had a home with a spare room. And so I went out of this lonely, empty house over the summer to stay with Howard and his family. And the other thing that I was very lucky, as I realized ultimately, initially, I felt a bit sad because I'd wanted to do NMR and thought that all mass spectrometry was adding numbers together and subtracting numbers, and that didn't seem to be terribly interesting. I couldn't have been more wrong. So Howard was developing new technology, and this was cutting edge, and no one else was doing it at the time. So basically what he was trying to do was to convert a mass spectrometer, which up until that point had been used by chemists and metallurgists and physicists, etc., to look at small volatile molecules largely. Howard had had the idea as a PhD student in Leeds that you could use mass spectrometry for sequencing peptides and proteins. This was absolutely unprecedented. So I had the tremendous privilege of being Howard's first PhD student, having the opportunity of being as it were, as a PhD student jointly with chemistry and the laboratory of molecular biology, which is why I'm now in the biosciences. Up until that point, I'd been entirely a chemist. I hadn't actually done biology very much prior to that. And it was a tremendous experience for me because it gave me the opportunity to work on completely new things. And one of the things that I worked on as a PhD student was trying to develop a technique called field desorption. Now, you don't need to know what that is, other than it is technically really quite challenging because it involves working with wires which are very fragile. They're about 10 microns in diameter, on which you need to grow carbon whiskers, so it finishes up looking a little bit like a bottle brush. And then you have to load the sample on the ends of these fragile carbon whiskers, insert that wire, as it's called, which is on the end of a probe. This is me, by the way, back in the mid-1970s. But on the end of a probe, which is inserted into the mass spectrometer, and the wire is then placed very, very close to a counter electrode. The wire has a voltage on it of about, or did have, of about 12 kilovolts relative to the earth of the extractor plate that was near it. So these whiskers were within a millimeter of that earth plate. And 99.9% of the time, what would happen as soon as you switch the voltage on, a whisker would fly off and short between the wire and the extractor plate, which meant that you then had to remove all of this, put it back on again, pump it all down, which would take about a day. So, not surprisingly, this did not become a major part of my PhD thesis. However, it was very important once the lab moved to Imperial. As Murray has already said, I was a postdoc here at Imperial, and I was very fortunate. Again, luck comes into it. I wasn't meant to come to Imperial. I was meant to go back home to Western Australia. The British Council had paid for a return airfare for me. So 3 years later, I went back home again and said, can I please have the nice job that I now am eligible for, since I have a PhD from Cambridge, to discover that Western Australia was in the depths of depression, rather like Britain in 2008 to 10, etc., because the mining industry had collapsed. And the mining industry was essentially the foundation of the West Australian economy. So fortunately, Howard had been invited to come to Imperial to the new Department of Biochemistry. It was new because prior to that, between the early 1960s and 1974, there were no undergraduate students in the biochemistry at Imperial. The department was effectively a research institute. So in 1974, Professor Brian Hartley joined from the Laboratory of Molecular Biology in Cambridge to reinvigorate biochemistry at Imperial. And Brian had a vision for the new biochemistry, molecular biology, mass spectrometry, all of these things, some of which were being developed in Cambridge and obviously elsewhere. And that's how I finished up coming here, because Howard Morris had applied for funding for new instrumentation, which he was designing or helping to design, which in particular had a mass spectrometer with a high-field magnet. And all you need to know about a high-field magnet is that if you have a high-field magnet, you can look at big molecules, or much bigger molecules than it had been possible before that. So that meant that essentially this technology was applicable to biopolymers, or at least smallish biopolymers. So what then happened was that I, fortunately, was offered a lectureship in 1979 at the end of my postdoc here. And obviously I had to think about what I was going to do as an independent lecturer as opposed to being Howard's postdoc. And that happened to coincide with people in other parts of the world knowing about the equipment that we had, knowing potentially what the technology could do. And the person who literally, in addition to Howard, who had obviously initially transformed my life, the person who then transformed my research life in terms of what I then subsequently did was the Professor Clinton Ballou of the biochemistry department, the chair of the biochemistry department at the University of Berkeley in California. Now, what was really sort of a bit scary for me at the time was that I got a letter from him. Remember, this was before faxes and you couldn't afford the telephone in those days, so letters came in the post. I got a letter from him asking whether he could come on sabbatical. And this was because one of his colleagues in Berkeley, Al Burlingame, who was then and still is one of the top mass spectrometrists in the world, Al Burlingame had been working on a whole variety of things for mass spec, including bio stuff, but one of the things he'd been working on were the moon rocks. His lab, he was at the Space, I don't know whether you can read this, Space Sciences Laboratory. So his lab had been analyzing moon rocks, and that meant that he got invites from the BBC to come to London to talk to the BBC for some of the programs, like the Horizon type programs that were being run by the BBC at that time. And one day he came to London, having heard about our mass specs, turned up on our doorstep with some samples from Clint Ballou. And Clint Ballou was working with sugars. So I managed to get field desorption data. Our lab at that time was pretty much the only lab in the world that could do field desorption, and certainly it was the only lab that could do field desorption on largish molecules. So Clint said, could he come on sabbatical? And he did. He came for about 4 months. It was amazing. This was the chair of biochemistry in Berkeley. He arrived, he put his lab coat on, and we went in the lab together. And he worked with me in the lab for the 3 months that he was here. That time happened to coincide with the development of another technology which came out of UMIST in Manchester, which replaced field desorption overnight. So all of the hard work of the 6 years or so that I had spent trying to get these whiskers to work was replaced overnight by a far more robust technology that used fast atoms or fast ions to ionize our molecules. But fortunately, that technology could be very easily retrofitted to the mass spectrometers. So by the time Clint arrived, which was a few months later, we had the technology in-house. And the very first molecule that I analyzed on it, you won't think this is special, but it is, this transformed the analysis of sugar-containing molecules, where all sorts of different glycoconjugates, literally overnight, could be analyzed using this technology. And very soon, it didn't need email and anything else for people to find out about these things. Very soon, there was a queue of people queuing up to come to Imperial. And they included, and some of their coming was because of other events. So, for example, Peter Albersheim here, who at that time was based at the University of Boulder in Colorado, was doing some very interesting work on plant cell walls, where he was identifying what he was calling oligosaccharides, which were effectively plant hormones, bits of the plant cell wall, the bits of sugar, which had profound effects on plant growth, on plant development, etc. And ICI Gellets Hill in the UK were very interested in these at that time, and they were funding Peter Albersheim. That's now, incidentally, Syngenta, if you know the name of the company. So Peter was often in the UK, and he had heard of this wonderful technology, and remarkably, he managed to discover my home address, even though I don't remember ever giving my home address to anybody other than family. And on a Sunday morning, there was a knock on the door, and he introduced himself. He had just come from Heathrow Airport. I was living quite close to Heathrow Airport at that time, with some samples in his hand, and he said, I hear you can do structural analysis of carbohydrate polymers. And I said, oh yeah, fine, okay, let's go into the lab. So we came into Imperial College, and for the first time in my life, the mass spectrometer decided to blow its main fuse. So I switched it on, and it went bang, and it didn't work again. Peter went away, and I thought, well, that's the end of him. But he left the samples, and I managed to get data for him. More or less at the same time, I had a visit from a scientist at the La Jolla Cancer Research Institute in Southern California. And he was interested in working with red blood cells and white blood cells and trying to understand their glycosylation. And the third person who came within sort of 6 months of each other came from Germany, Heinz Egger, who went on to become Chancellor of the University of Bonn. And Heinz was working on human milk sugars, in fact, 30 years before his time, because it's now very, very trendy to study the sugars in human milk, because it's known that they're really, really important now with respect to the development of the baby. So within a few months, it was possible to get data on these projects and on others. And within a couple of years, we were starting to work with a whole variety of people around the world. Now, I still had a relatively small team at that time. There were 2 PhD students who'd started in 1981 and finished in 1984, had been helping with the majority of these projects. The other thing I should say, which I feel very grateful for, and I do sometimes feel it's not quite so good for the newer generation, is that we did have time to work in the lab. I worked full-time in the lab through the whole of the 1980s pretty much, apart from teaching. And obviously teaching is roughly a third, was then, still is a third of most people's lives as academics. But the rest of the time was almost entirely in the lab. The other wonderful thing that happened in 1984, of course, that milestone year for a lot of people who grew up in the 50s, 60s, and 70s, who had read the 1984 book and thought all sorts of horrible things were going to happen by 1984. My daughter was born, and I had been invited about a year earlier to give a plenary lecture on this new technology at the International Carbohydrate Symposium. So that international meeting was every 2 years, and to be a plenary lecturer was a pretty important invitation, because you only got one invitation to be a plenary speaker at this meeting in those days. So I had agreed to it, but the timing was rather bad. So Emma was born in May, and the meeting was in Utrecht the first week of July. So I flew over to Utrecht with her, and at the meeting, it was wonderful, because everybody was great. In fact, Peter Albersheim, people still tell me about this, that they couldn't believe the fact that he happily gave Emma the bottle while I ran a workshop that he'd organized on field desorption and fast atom bombardment. So that was a pretty important time for me, and I just want to sort of highlight how important, and I'll come back to that at the end, the team has been. I think being part of a research team and being able to work alongside PhD students and postdocs is actually a wonderful way to spend one's life. So this is daughter at the age of about 7. So, and these are some of the collaborators at that time. So I hopefully have given you a glimpse into why sugars are important, but I just want to tell you a little bit more about them. So these are the symbols that I'm going to be using on the slides in a moment. And I just want to highlight the fact that sugars are not really that complicated for those who think they are. So essentially on this slide is the repertoire of sugars that are important in us humans on all of our glycoproteins. And they are all pretty much related to each other. There are 2 very important sugars, the diamond and the triangle, that we'll come back to in a little while. So sugars are everywhere. These kind of sugars are on all our cell surfaces, and you can see them in textbook pictures, often referred to as the glycocalyx. What do they do there? We don't know for many of them, but there is compelling evidence now from labs over the last 20, 30 years that they are involved, at least as one of their roles, in recognition processes. So, for example, when viruses or bacteria infect a cell, the initial stage of the infection, the engagement of the 2 cells, involves sugars on one of the cells being recognized by binding to a sugar binding protein, glycan binding protein on the other cell, that is usually referred to as a lectin. And one of the systems that has been studied in greatest detail with respect to this recognition is the trafficking of white blood cells. So way back when I first started working with Minoru Fukuda from the La Jolla Cancer Research Institute, we started to look at the kinds of glycans that were expressed on the surface of white blood cells, and we were particularly interested in knowing how they changed in, for example, leukemia. And one of the earlier structures we identified, which was completely novel for this kind of system, had only been seen once before in a rather obscure glycoconjugate in the brain, was this structure here that is called SALA-LUSX. And importantly, it's got these 2 rather important sugars on it, the sialic acid and the fucase. About 6 or 7 years later, the lectins that recognized that structure were characterized. They were called the Selectins, and there is now a mass. In fact, I had a quick look on PubMed this morning. I think there was sort of like 20 or 30,000 papers since the 1990s on this kind of area of biology, not just glycobiology. So the key thing is that the cluster of sugars is recognized by the lectin, and that results in the white blood cell rolling over the surface of the endothelial layer, spreading out eventually, and then squeezing between the endothelial cells as it traffics out into the tissue. Now, there are obvious applications of that observation, and in fact, the discovery of the selectins resulted in a flurry of biotech activity in the 1990s. Most of the biotech companies that were set up in the early 90s collapsed in a heap within 6-7 years, but that's normal. A few of them stayed, and one of them is this one here. And this company, if you see the date there, that's last month, has just announced the results from the phase 1 clinical trials of a chemically modified mimic for the treatment of acute myelogenous leukemia. So that's roughly the time that one would expect for going from original discoveries to the translation. So what do we do most of the time? Most of the time what we're doing is we're analyzing these glycans to try to understand structure function. And one of the things that the group optimized during the 1990s was being able to do that on more or less anything. You could take a little bit of tissue, or you could take like a Drosophila lava, or you could take a band off a gel. It didn't matter what the sample was. As long as it was possible to make a pool of glycans, then those could be analyzed by the technique that we call glycomics. Now, for you to understand a little bit about what I'm going to show you, I do need to give you one little bit of, as it were, undergraduate glycobiology biochemistry, which is what happens to a glycoprotein when the glycan is first added onto it, and how is that glycan then manipulated in the biosynthetic process. And what is very interesting about glycosylation of proteins is that the first step of the glycosylation is totally conserved throughout evolution. So it doesn't matter what kind of cell it is, as long as it's a eukaryotic cell, i.e. there has to be an endoplasmic reticulum for it. So what happens is there's this precursor glycan, same symbols that I used before, which is then modified. So essentially it goes through a production line of being trimmed down and then built back up again. And what happens by the time you get to the end is that whereas it was homogeneous at the beginning, because there was this one glycan, by the time you get to the end, it's a heterogeneous mixture, which varies in the amount of manipulation, if you like, of the glycan in that pathway. And this is why many people hate glycosylation. They don't want to have anything to do with it because they think it's a nasty, horrible mixture, therefore you can't work with it, therefore you can't understand anything. It couldn't be further from the truth. You can work with it. And in addition to that, it's not a nasty, horrible mixture. It is absolutely precisely fine-tuned by the biosynthetic machinery. So, in other words, glycosylation is incredibly sensitive to this biosynthetic machinery. And if the physiology and metabolism, as it were, within the cell changes, so will the glycosylation. So what kinds of glycans do we see? Well, these are the antenna. The glycans are described as having a core and then with antenna on them. These are the typical antenna, and all you need to really know is that you actually know a lot about them. So they include, for example, the ABO blood group system. They also include the other blood group system, which, again, most people don't know about because it's not critical for blood transfusions, the so-called Lewis blood group system. And what I think you hopefully see from these patterns is that basically it's a bit like a very limited repertoire of Lego bricks. You have your yellow circle and your blue square. If you want to make your antenna longer, you simply randomly repeat them, and then they get decorated with these diamonds and triangles. And those are what are absolutely critical to the biology. So the glycans are looking like this, and what we can use our mass spectrometer for is to tell us how many antennae, how long the antennae are, and what are the decorations. And then that information can then be used to try to understand the biology. Now, in recognition of the help that that kind of information can give to the bioscience community, glycomics analysis was one of the infrastructures that was funded by the National Institutes of Health when they set up the Consortium for Functional Glycomics in 2001, with the aim of the consortium being to provide resources to the international community for free, which they did for 10 years. And the analytical core of the CFG was based here at Imperial. And the reason for that was because of our reputation for being able to do this, which they tried to duplicate at UCSD and were unsuccessful. So they tried it there for about a year, and then they realized it's not the technology that's important. Obviously it is, but the fundamental thing that's important is the skill and the understanding of what to do, which of course we had here because we'd been working for so long. So this kind of data is on the CFG's website for different types of cells like neutrophils, mast cells, eosinophils, where it is very easy to rapidly identify by looking at the website what the main kinds of sugar epitopes are on these different cells. So let me just give you an example now of how you can use that information. And I'm not going to tell you a complete story because it's not complete yet. It's a half-finished story, but it's at a pretty exciting stage. And this comes out of work that was initiated with us by Gary Clark here back in the early 1990s, when Gary was interested in trying to understand why it is that in the human reproductive tract, and obviously other animals as well who are similar to humans in that they have placental pregnancies, why it is that the mother does not reject the fetus. In other words, if you think about the placenta, it implants into the womb, or it grows, obviously, as after the embryo implants, but half of it is foreign because it's expressing the father's antigens. And of course, with modern in vitro techniques, the whole of the placenta and the developing embryo can be foreign to the mother in the case, for example, of an egg donor. And this is a question that was posed by the Nobel Prize winner Peter Medawar, who worked at the National Institute of Medical Research at Mill Hill, leaving aside his use of the, pregnant mother itself, the use of itself for a pregnant mother. It was a really, really important question to be posed, and it has still not been answered today. I'm not saying it hasn't been partially answered, but glycosylation almost certainly plays a central role. I can't say it certainly does because there's no experiments that can directly prove it. The hypothesis of what glycosylation might do was articulated by Gary in this so-called HUFEDS, because everybody likes to have an acronym. Obviously, it wasn't only him doing this kind of work, where his hypothesis was that the cells, or the gametes, the sperm and the egg, are expressing glycans, and there will be lectins being expressed by other partners. Once the embryo starts to develop, obviously it develops from the egg, there will again be glycans being expressed on the embryo, and there will be recognition processes taking place where there is communication between the developing embryo and the immune system of self, non-self recognition. And what Gary was essentially saying is that the processes that are occurring for immune suppression in the reproductive tract will be mimicking processes that are, as it were, generic to the immune system. So I already told you earlier that slalyl X is that critical epitope on white blood cells and enhanced in cancer. It's involved in white blood cell trafficking. The prediction from this hypothesis back in the 1990s was that either the same structure or a similar type of structure would be being expressed in the reproductive tract. Now, it took us almost 20 years to actually prove this for the human egg. This involved a team of people, Gary and people at Academia Sinica. This was a PhD student, Kei-Hui Ku was a PhD student in my lab in the early 1990s and now has his own independent lab, obviously, in Taiwan, Imperial College people. And the material, the egg material, was obtained from the University of Hong Kong. This wasn't easy to do. We worked with 195 unfertilized human eggs. This is meant to be a full stop here, just to give you an idea of how big a human egg is. Okay, it's roughly, if you draw with a pen, a full stop on the page. And we had to use that small amount of material to effectively do all of our detailed analysis so that we could be sure we knew what we were looking at. I thought I should show a tiny bit of data, even though you don't need to fully understand it. Hopefully you'll understand these patterns here and the fact that we have a spectrum where the masses are telling us about what those sugars are. If I come on to the next slide, you can see them in a little more detail. What was absolutely stunning to us was that pretty much every single antenna is terminated in slalyl Lewis X. The abundance of slalyl Lewis X on the surface of, not of the human egg itself, because it's inside a gelatinous coat that's called the zona pellucida, but the zona pellucida is what the sperm recognizes, absolutely coated with slalyl Lewis X. And you can see that if you use an antibody. The red is lighting up the egg. And you can see it if you understand this kind of picture, that an antibody to slalyl Lewis X will inhibit sperm-egg binding. But notice it doesn't inhibit it to 100%. This is very reproducible. So it's not as simple as just slalyl Lewis X on its own. And we still don't actually understand exactly what it is on the human egg that is being recognized by the sperm. Now, you might well say, well, what about sperm? You need immune suppression for the sperm as well, because after all, the woman's immune system will see it as foreign, and it's got sort of 2 days to kill it off normally before the sperm manages to get to the egg to fertilize it. So again, the hypothesis is that the sperm should be expressing glycans that anywhere else in the body would be recognized as being either perhaps foreign or certainly involved in immune suppression. And that turns out to be the case. So if you look at human sperm and look at its glycans, they are absolutely jam-packed full of an epitope which is called Lewis Y, which has 2 of these triangles on the antenna. Now, the interesting thing about Lewis Y is it's highly expressed in some parasites, particularly schistosomes, or I should say it's a slightly different isomer of Lewis Y, but effectively in terms of its 3D structure, it's Lewis Y. And there is a lectin on, for example, immune cells, the dendritic cells, which are able to engage with these kind of structures. So the hypothesis then essentially is that we have these glycans that engage with lectins and dampen down the immune system. And many of them are being expressed at very high levels on cancers. So Lewis Y, for example, in women is regarded as a cancer antigen because we don't really express it anywhere in any abundance. Of course, it's very different for you guys in the audience. If you look at something like pancreatic cancer, for those who are not familiar with this kind of staining, essentially everything that's being stained inside here is being stained to recognize Lewis Y. So again, the hypothesis being that it's mediating immune tolerance. So the current state of thought is essentially encapsulated in this slide here, that we have a problem with respect to immune suppression. You absolutely have to have it in order to survive as a human race, if you're going to have placental pregnancy and you're going to have the baby being essentially carried to term over a long period of time. But of course, that leaves us vulnerable to the fact that exactly that same immune suppression system can be, if you like, hijacked by cancer cells, hijacked by parasites, although very often parasites like schistosomes don't kill people off, they just make them feel pretty sick for 20 years. So, in principle, depressing, but actually I would say it isn't, because if we can understand this properly, and of course, currently, and it's all over the newspapers, T-cell therapy is becoming one of the hottest topics in medicine. If we can really understand what's happening in these immune situations, then it should be possible to actually hijack the system ourselves. In other words, to be essentially making the cancer vulnerable to the immune system by manipulating the glycans that are on the cancer. And Gary Clark, my collaborator in the US, is already doing animal experiments that are suggesting that that may well be a way, not for treating all cancers, but certainly treating some. So I said at the beginning I wanted to tell you about immortality. I just thought, in view of the fact you might be feeling all depressed about the fact that we're all going to die of cancer eventually, because you know, we're programmed to it, I thought I would show you how glycans can help us be immortal. And so I just have a couple of slides here on a project with a collaborator in India. Babu came here actually on a travel award for a fellow a few years ago, well, actually more than a few years ago, when Tony Blair signed an agreement with India for exchange of scientists, one of the first exchanges that was implemented. Babu succeeded in getting one of the awards. So he came to learn about glycomics, spent a year in our lab, then went to the US, and now has his lab back in India. And he's very interested in hydra. Now, the thing about hydra, if you don't know, is that they're really remarkable creatures. You can cut the hydra head off and it will grow another head. But even more sort of cool than that, you can take your hydra and you can sonicate it until all it is are individual cells, and then you wait a little while and it reaggregates and becomes a hydra again. So what Babu and his colleagues found was that you could prevent that happening if you used a lectin that recognized that triangle that I showed you on all of the other pictures, that's fucose. So here's the development from, these are the cells aggregating, this is normally, and this is the hydra that we're seeing up here. So this is what happens to the cells if you then add into the solution that these cells are growing in, or the media they're growing in, if you add a lectin that blocks fucose, that inhibits fucose, a fucose-binding lectin. Over the same period of time, these are two different lectins. You can see the cells are just an aggregate. They have no longer, are they able to form the lovely hydra shape. What we found, we're absolutely stunned by, is that the hydra are expressing masses and masses of fucose. So these were unprecedented structures with respect to the amount of fucose. The only time that any structure like this had been seen previously was in work that we'd done back in the 1990s on schistosomes. So schistosomes also are able to express these oligofucosylated antigens. So what we hope is that if we can better understand these processes, we can harness them to our advantage. And for anyone who might be interested in the hydra, I just want to point out that not only can you use, as it were, a lectin that is a tool, which is what was used on that earlier slide, but actually the hydra themselves, Babu has now shown, are expressing a fucose-binding lectin, so that you can start to hand-wave and hypothesize about how the glycans on one of the cells, say over here, is being bound by the lectin on the other, or perhaps even on the same cell itself, and signaling in some way inside for that development process to take place. So for those of you who've all been asleep for the last sort of 50 minutes, this is just telling you what I've talked about, as opposed to what I was planning to talk about. And I just want to finish by showing you the press release for the NIH Consortium for Functional Glycomics Award. They were called glue grants, actually, sticking people together grants. So I'll let you read that. It's a typical over-the-top press release, but I wouldn't like to say that it isn't true. I mean, glycans, sugars are really, really important to absolutely everything. So I'd just like to finish again, coming back to what I said at the beginning. None of this would happen without all the people who have worked with me over the years. And of course, I couldn't put up pictures of everybody. So these are the pictures of current people. And I particularly want to acknowledge Howard Morris, without whom I wouldn't have done anything, or I would have done a PhD, but I'd have gone back to Australia and probably just stayed at home raising kids and whatever, wouldn't have been a professional person. The other two people I want to particularly flag up are Maria Panico, who has been an absolutely superb colleague in the lab and associated with running all of our projects over quite a long period of time. I won't say how long in case she gets upset. But Maria, without her, we would not be doing all of the really advanced mass spectrometry that we're doing. And Stuart Haslam, who joined the lab in the 90s, and of course is now an independent reader in the department, taking forward his main interests, which are very much on the side of parasites, immunology, etc. And finally, of course, as I mentioned, we've had lots and lots and lots of collaborators around the world, and there were too many to name on the slide, but I just thought I would highlight the fact that on this slide here, which is the National Happiness Rankings, this is for true, I took this off the web, See, Australia here is actually a little bit happier than we are in the UK, so maybe I should be heading back to Australia. And thank you for your attention.
I'd like to thank Anne for that lecture. I think it's time for a few questions. These could be.
Whether you're aware of that, and Silwood may want to ask questions, and if so, they're going to put it onto the computer. So, anyway, I'm happy to answer questions.
So, the little bit I know of evolutionary biology tells me that males should not be subjected to the immunosuppressant efficacy of Lewis Y. Is that the case? Because we don't make babies. So why would...
Yeah, but your sperm will be killed off by us. No, I mean, that's a really important point. The sperm need to be protected, because otherwise, how are they going to survive in a very hostile environment? There is a very active immune system in the female.
It's the immunosuppressive system that needs to be silenced by our sperm, right?
Sorry, I missed the beginning of that.
It's the female's immunosuppressed immune system that needs to be suppressed by the sperm. It's not the other way around. So we could cope in theory without the immunosuppressive capabilities of the Y.
Well, before the egg is fertilized, there is a very active immune system in the woman's reproductive tract to ensure that no infections occur, or infections are minimal. So the immune system for killing the infective agents will, in principle, act against sperm as well. But I have to say that I'm the analytical person, so I am skating on thin ice, if anyone knows, as I'm sure they do, a lot more about the immunology than I do. But you're right overall. It's only that tiny little window of time that the sperm need to be worrying about an immune system. Yeah.
I was wondering, because we managed to implant eggs from other women into women. So theoretically, an egg would not need an immunosuppressant because it is within its host.
At the stage of the egg, I agree, yes. Yes.
So why can we implant a different female's egg into another woman?
Well, our evidence to date is that the glycosylation isn't different on the actual egg between women. So that almost certainly is going to be a preserved structure. What will then happen as the embryo develops and as the placenta develops is there's a much richer repertoire of glycans, and that's when you can start to get the differences.
Any other questions? Okay, then so we'll have tea and coffee outside, and I'd just like to thank Anne again for fascinating lecture.